Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Sarah Baatout. She's the director of the radiobiology unit at the Belgian Nuclear Research Centre in Belgium. She's also a guest professor at Ghent University, and we're going to talk about space exploration and protecting astronauts from space radiation, I believe. So, Sarah, thanks for coming.
Thank you very much. Thank you for inviting me.
Well, tell me about your work in regards to space and in regards to radiobiology. What have you been studying?
Okay, so as you mentioned, I'm a radiobiologist or radiation biologist, and I'm investigating the impact of radiation, ionizing radiation on health, in order to understand how we can take advantage of radiation, how we can improve radiotherapy treatment, develop new radiopharmaceuticals, and predict better the risks induced by radiation, by exposure to radiation in each individuals. So that can be patients, but it can also be astronauts.
No, that's excellent. Just living on Earth, what kind of radiation are people exposed to on a day-to-day basis?
On a daily basis, the dose of radiation that reaches the Earth is quite small, is very minimal, with no impact on health, because on Earth we are really cocooned and very well protected by our magnetic field and by our atmosphere. So that helps us also to not get exposed to radiation. But the situation is very different in medical exposure. After medical exposure, if you want to treat a patient with radiation, kill cancer cells, then you need, of course, much higher doses that you can give in the medical context. And in space, on the ISS, on the International Space Station, astronauts are exposed to much higher doses because the dose of radiation increases with altitude. So the higher you are, the higher dose of radiation. And so on the ISS, on the International Space Station, you have doses that are about 150 to 200 times higher than what we encounter on Earth.
Oh, wow.
Yes, so it's much higher, and it's even worse if we think about the future scenario that we are preparing, which are going to the moon, going back to the moon, having a moon village there, and later on going to Mars. There, the doses of irradiation will be even much higher, 300 times, 400 times higher, going to the moon and back, and then even 1,000 times higher, going to Mars and back. So there is really a lot to do to prepare astronauts to face this.
Well, so when people went to the moon, in '69, was it just that they were exposed to much higher levels of radiation for, the whole journey, or is it that in certain areas of space outside the Earth, the radiation goes up tremendously?
They were exposed to, of course, higher doses of radiation than we have on Earth, but for a very limited amount of days, because they only went for a few days and then came back. Even if some of them performed what we call extravehicular activities. Extravehicular activities is when they went outside the space vehicle or went on the moon surface to take some samples and take some pictures. And so they were a lot less protected because they only had their spacesuit as only protection. So, but the dose that they received was for a short period of times, which was not such an issue. The future human space exploration program, which is an international program where NASA is involved, in which NASA is involved as well as the European Space Agency and the Canadian Space Agency and the Russians. The idea is really to go back to the moon and stay there for quite a certain number of months, which means that you have not only the mission, the traveling to the moon, but also staying there with the protection that you can get from the settlement there, the protection from the habitat, and from the spacesuit.
What about an intermediate step of flying in a plane? How much radiation would someone be exposed to versus being on the ground on Earth?
Yes, so indeed, pilots and crew, air crews receive, of course, higher doses of radiation. Certainly, when they fly, for example, above the Atlantic at a very high altitude, the dose of radiation is a little bit higher, but still, it's like the dose of a dental radiography, which is pretty small. Pretty low dose of radiation. So one trip over the Atlantic is not harmful, but air crews are really monitored. So they have what we call a dosimeter, and this dosimeter record the dose of radiation that they are exposed to. And so they are only entitled to a certain dose per month because they are working their job. So it's considered as an occupational exposure to radiation. And so this is regulated. There are laws in terms of the number of those that you can get. And so they have a specific program of exposure, and they can't go over this type of and a certain level of exposure of radiation. But it would be much more to go to the moon. We are really talking factors of 200-300 times higher doses, and for a longer period of time. So in that way, it's very important to protect the astronauts. So to develop programs first physically to develop the best shielding, radiation shielding that we can imagine and test in order to make sure that they are sufficiently protected. And then humanly, in terms of what we can do at the level of the human body, there are different ways, or using some type of radiation protectants. So these are type of molecules that will be able to stop somehow the damage induced by cosmic radiation, and or will boost the way the cells are repairing the DNA damage, so that will induce a better repair, less damage. And you can also do different types of studies that we are performing for the moment on the International Space Station. And these are studies in order to understand what is the sensitivity of each astronaut. So for that, we get the blood samples from astronauts before they leave to the space station. And when they come back, and we study how their machinery, their DNA repair machinery function, if it functions better or less well, and what we can do when we have analyzed, because biologists, of course, want to understand.
Well, what does that mean, the DNA repairability? How do you quantify that?
So basically, the type of radiation that astronauts are exposed to is quite mixed. It's not just one type of radiation, because on the ISS you have solar wind, which are electrons and protons and alpha particles. It's a very mixed exposure. And you have also high energy protons coming from the solar particle events that can come, which is very intermittent. It's not all continuous. And you have then the galactic cosmic radiation, which are high energy protons and alpha particles. And so this mixture of radiation that you have in space will induce what we call very highly complex DNA damage. And this complex DNA damage means that the DNA is broken, will be broken in different places and quite close to each other, because in fact this type of irradiation will induce along the track of irradiation quite a lot of concentrated damage in the DNA. And this we know that for the cells to repair this DNA, so because these radiation are very dense and they really ionize along the track in the DNA, and they cause unique damage to all the biomolecules, the cells and the tissues that they will encounter in the human body. And so our work is really to understand what is the distinct pattern of this DNA damage, and what is the profile of the oxidation, because basically this DNA damage is a kind of oxidation, a very corrosive way of attacking the DNA. And is our body going to be able to repair at the same speed? So that's why we study these astronauts, and we study how powerful, how efficient their system is, and in their blood, because the blood is quite sensitive to radiation. In fact, the immune system in general, there are quite different parts of the human body that are sensitive to radiation and to space conditions that make, in fact, the astronauts weaker in terms of health status in general.
Well, all right. How do you quantify this? How much weaker? How do you know when?
Yes. Okay.
Based on a given amount of radiation, does it go by body weight? Like, how do you know what's going to happen to somebody?
So you can measure different types of parameters. So if you look, for example, at the impact of space conditions at the level of the immune system, so you collect blood and you measure different types of proteins. So in our genome, when the genome is attacked, so when it's hit by radiation, it will spontaneously want to start to repair the DNA damage. This is a very natural way of the cell. And for that, because our DNA is extremely condensed, so it's very condensed, to let this repair take place, the cells and the DNA needs to decondense and to open up in order to let the proteins, the repair proteins, to start to repair any type of DNA damage. So it can be big breakage, it can be a double strain break, which is like the two strand of DNA being broken, or a single strand break, or a base damage, or a very small damage, or even bigger damage, or a whole chromosome which is cut. So they can be massive damage. And so you measure different types of proteins that are the ones that are kind of firemen within the cells, that are going to stick to the DNA where it's damaged, and you can quantify that. You can use, for example, an antibody that will stick to every places where the DNA has been damaged. But you can also measure the full genome and the full transcriptomics, so the gene expression of the cells, and then come up with something which is much more precise, which is an idea of the gene, of the concomitant gene expression of about 20,000, 30,000 genes at the same time. And there you get a very precise idea of how each body, how each astronaut is reacting towards radiation. And with that, you can determine the radiation sensitivity of the astronauts. And this is very important for the future mission to the moon and to Mars. Why? Because you, through this specific radiation sensitivity of the astronauts, because in fact, when you look and when we are facing radiation, we are not all equal. Some of us will be very radiation sensitive. Some others will be very resistant towards radiation. And we see that in patients, and it's the same in astronauts. So some of them are more sensitive than others towards radiation. So it means that they will repair less.
What does that mean, sensitive? Like, how do you know how sensitive someone is? I thought the radiation takes quite a long time to show up in terms of ill effects. Like, how do you quantify this?
You are totally right. So you can have some impacts that come years after exposure to radiation, but you can also monitor impact just after exposure and see what is the concentration and the amount of these DNA repair proteins, how efficient are they, how active they are. So all that will play a role in this radiation sensitivity. But also it depends on our genetic background. So each of us has got a certain pattern of mutations that we have that we receive from our ancestors, and on top of that we have the changes that occurred from birth onwards. So all the epigenetic changes that has taken place and that will stimulate, boost some gene expression or inhibit some other gene expression. So all that will play a role in the generic individual susceptibility of each astronaut. And knowing that in advance, before the astronauts fly, would be very interesting because you can then develop what we call precision medicine of astronauts in order to determine for them what would be the best program for radiation exposure, taking into account their specific sensitivity to radiation.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
And what have you learned from medical radiation that people get, here on Earth? Like, what kind of treatments where people get, radionuclides, and what's some of the interesting facts about that?
Some very interesting and surprising insight is first how complex things are, in a way that not only we have in our genome, we have, of course, tens of thousands of genes that are each kind of playing a certain role, but also radiation has got a different impact on different cells in different tissues. So you have parts of the body that are quite sensitive to radiation. For example, if you look at the lens, through exposure to radiation, you might have an induced risk of developing lens opacity that we also call cataract. So this is one of the tissue which is quite sensitive to radiation. But there are other tissues, like, for example, the cardiovascular system. Some of the cardiovascular cells are sensitive. The immune cells, lymphocytes, are sensitive. The reproductive system is very sensitive to radiation. So all these systems should be certainly better protected. But what is also interesting is that so the genes are expressed differently and respond differently in the different tissues. They also respond differently depending on the type of radiation. But it's possible. What is really interesting that what we have been developing is possible to develop biomarkers, and biomarkers really of radiation response that can predict, and that really goes towards precision medicine for patients, that we can really implement into the clinics through the use of blood, but also what we have developed and what we are currently developing is through the use of saliva. So with a non-invasive material, biological fluid, we can determine the exposed dose and determine the risk for the patient. And that can have an important impact on the radiotherapy treatment. So radiotherapy treatment is, at the day of today, usually the same type of radiation treatment depending on the type of cancer, for example. So you will get a certain number of fractions of radiation for a certain number of days, depending on the type of cancer.
How much do the effects on the targeted cells versus the rest of the cells in the person's whole system? How is that quantified?
Yeah, absolutely. So it's quantified clinically. It's quantified by the fact that healthy cells around the tumor will repair much faster when they are irradiated than cancer cells. So this is really one important fact that basically will help to kill the cells. Also, radiation that it's given through external radiation, like conventional radiotherapy that is used, like will be targeting the tumor. So you give a very high dose at the level of the tumor, and you spare the healthy tissue around. So the dose around is supposed to be much less, but sometimes you get still a certain number of dose, a certain level of dose on the surrounding tissue. And it's important to basically understand what is the impact on the healthy tissue, how we can protect this healthy tissue, developing radioprotectants, but also protocols, radiotherapy protocols, in order to target better. So there are different ways. For example, we learned and we discovered that we can, by using photons or using not the conventional radiotherapy, but using carbon ions, you can really kill much better tumor cells, and you spare the healthy tissue also much better. And not only that you kill better the tumor cells, but you inhibit totally the risk of tumor cells to develop metastasis. We managed to show basically that carbon ions that we use for hadron therapy can really help to decrease the risk of metastasis and really induce much higher effect on cell killing. And we are also developing now more and more radiopharmaceuticals, which are a combination of radionuclides, so radioactive molecules, coupled with a vector, a kind of a linker, and which is attached to a certain type of biomarker, a type of molecule that will target and that will attach specifically to the cancer cells. And there we see that we really also improve the treatment and also target not only the primary tumor, the primary cancer, but also any type of metastasis that we would have in the body that would not have been detected.
Can you shield someone when they're getting radiation? I guess if it's pinpoint radiation, that's one way, but if they're taking a radioactive pill, I guess there is no shielding at that point, right?
Indeed. So there you have indeed, and then you need to have a radionuclide, a radiopharmaceutical, which is very specific to the tumor you want to target, and that it's not that it doesn't start to spread in the whole body and start to stick at different tissues and healthy tissue that you don't want it to stick to. So it needs to be for that you need to really work on the best molecules first, which is very specific to the tumor that you want to target and to treat. And but indeed, there are studies that are taking place and that we are performing in order to make sure that the dose will reach the tumor, will not start to spread in the rest of the body, and that dose, which is the radionuclide, which is not absorbed, will be directly eliminated through the kidney and then in the urine. So you need to make sure that you don't have an aspecific targeting, and this is very important. But for the moment, I would say that it's really having a boost to develop new radiopharmaceuticals, because it's important to go in different ways and going towards external radiotherapy treatment with conventional radiotherapy, even improving personalized medicine for those patients, but also developing new radiopharmaceuticals to treat cancer that we are not able to treat well at the day of today. So there is really a really beautiful future for that as well. And all this research is really going hand in hand with space research, because basically everything we understand from space, type of radiation, the mixture of radiation, and the harmful effects of space radiation is very interesting in terms of research, because it helps us to take out these characteristics, these harmful characteristics, and to implement them in the clinics and to improve our radiotherapy protocol. So there is really a win-win performing space radiation and improving radiotherapy treatment, because they go somehow hand in hand, and they kind of, the type of technologies and the type of radiobiology is quite similar for both disciplines.
So what's some common shielding used for radiation externally?
So if you are on Earth, you can use lead, and which is the common lead jacket, which is the common protection that you would use in hospitals, in the clinics. But lead is very heavy, so every kilo to go to the moon and to take on board on the spaceship would be extremely expensive. So we are looking at other possibilities and other ways. And there for space, we are really looking at very small atoms like hydrogen or helium, very small atom, the smallest atom, or boron. And those are very good protectants. So you can imagine, for example, a shielding jacket that are made of polyethylene. Polyethylene is a long molecule of carbon with a lot of hydrogen on each side of the carbon. You have a lot of hydrogen, and this hydrogen can really protect against space radiation. Water is also a very good protectant. So if you look at the day of today on the International Space Station, you have 3 parts of the space station that are very well protected, where astronauts can reach and can go very quickly in case of a solar flare, because in case of a solar flare, they have a few minutes to reach one of these 3 protected areas. And one is the water is next to the water tank, so where the water supply is stored. And the two other places, one is in the lab, and one is is the sleep place, the sleep compartment, where basically they are protected with this polyethylene as well as in the lab. So this is for the moment what is used on board of the ISS. And for the future mission to the moon, right now we have polyethylene. There is a jacket. The jacket for the next missions would be in polyethylene. But there are other components, and amongst the studies that we are performing, we're also studying the spacesuit. How can we make the spacesuit the most radiation resistant, but also to have some kind of detectors that can monitor radiation dose in an active way, so continuously, and give a certain alarm in the case of reaching a certain threshold of radiation dose. And so we are studying different types of components and molecules that would protect the astronauts the best inside this spacesuit.
Well, what new materials do you think will take the place of lead? What's your guess?
Oh, there are different types of material. There is a lot of research performed on really small molecule with hydrogen, with helium, a lot on boron, and a lot on polyethylene as well. For the moment, this is what is taking place. But I think what we are also currently developing is trying to have a biomarker chip. So to really have a chip that could give an idea very quickly for the patients, but also for astronauts in the future on the type of dose, but certainly also the type of damage that they are encountering inside their body. And that's really important, certainly in the clinic, because it would help to stratify patients, to stratify and to fine tune and to adapt the treatment for these patients. I think to me also what is really the future is coming also from the theranostics. So these are radiopharmaceuticals for which you could do and that you could use and for diagnostic and for treatment. So that would be very helpful to be able to do and to diagnose the disease and to treat at the same time. So there is really a lot going on for the moment in that. And then for human space exploration, the future is really also developing the best medical kit, so the best technologies for diagnostic of all type of diseases that could be encountered by the astronauts in space, and to be able to treat in space on board, because a mission to Mars and back would last for a few years. We speak about 2.5, 3 years with the technologies of today, with not possibility to come back. So we really need to take on board everything we can think of. So developing the tools, the technologies, the pharmaca for preventing and treating any type of disease that could be encountered.
So if you look at the space journey in terms of like number of days of travel, what's the current upper limit of safety because of the radiation?
So for the moment, we are really, there are a lot of discussions between the different international space agencies. NASA and European Space Agency, as well as the Japanese, the Canadian, the Russian, in order to really decide internationally what would be the limit. And because for the moment, each space agency has got different types of limits and threshold that they would tolerate in terms of a whole exposure during your whole career as an astronaut. And but this endeavor to go back to the moon and going back and going to Mars, it's really an international endeavor with astronauts from different countries and different space agencies. So there is really a need to find and to harmonize the threshold that we will tolerate for this long mission. For the moment, even if we are, even if an astronaut stays a few months on the ISS, on the International Space Station, the dose that is reached is still okay, is still under the level and the threshold limit that is acceptable. But when going to the moon, staying on the moon for a few months, or going to Mars, this threshold has got to be rediscussed and maybe revised. So this is taking place for the moment between the different international space agencies.
And I guess it's, you can shield someone inside of a ship, but like you said, if they go outside the ship to do a repair or whatever they do, then the radiation gets tremendously higher, right?
Exactly. So it gets much, much higher. But what we are preparing is when going back to the moon, it will be not just one extravehicular activity. It will not be just one time outside, but we speak about going about 70 times outside the moon village and then going for sampling, for exploration on the surface of the moon. So it means 70 times going outside, which much higher dose of radiation, with a mixture of exposure and mixture of radiation, which are difficult for the body to repair. So the risk will be much higher going outside and on these extravehicular activities. And so therefore there is really a need for more research on the shielding in the space habitat, but also on the spacesuit concomitantly and in parallel.
So how far away are any of these innovations, do you think? I mean, when are they needed? Like when are people planning to go back to the moon and spend any significant time there, or even go back, period,
For the moment, the planning is that humans would go back to the moon, really on the moon, from 2030. I think end of 2020, start of 2030s is what is in the mind now and what is foreseen. And then maybe 5 to 10 years later to go to Mars. So technologies are being developed at a very high speed. So technologies, not all technologies are developed, but quite some are already available on Earth, but would need to be miniaturized, would need to be simplified to be able to work in space. And also they need to be adapted for working in microgravity. So where everything is floating. So if you have liquid inside an equipment, a machine, so then this liquid can become a hazard because it can start to float and leak. So you need to, every machine needs to be adapted to be able to be used in space conditions. But research is going quite fast and quite well, and with a lot of collaboration at the international level. So I think we can reach this target of for going to the moon.
Well, very good. Sarah, what's the best way for people to find out more information about your work?
I think the first place where I would go is our website. We have a very good website, sckcen.be, where you have quite a lot of information. Then we're also quite active on the social media. So I would just go on Twitter and LinkedIn and Facebook if you are interested. Also, we publish quite a lot of review articles on these thematics that are quite understandable, we think, for everyone, and they are open. They have an open access. Maybe another way also is to follow a TEDx conference that I gave not such a long time ago. But I think also for anyone interested in this kind of topic, just listen or read to general media related to sciences and to medicine, to radiotherapy. There are really a lot of information that you can find on general websites. And I'm sure with also what you are doing, your type of podcast, that's really helpful as well to reach a big audience and for everyone to understand better science and medicine.
Well, very good. Well, Sarah, thank you for coming on the podcast. I appreciate it.
Thank you. Thank you so much for having me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.